I got it.
There'll probably be more people that show up. Like a lot of people don't tune in at exactly 7.
Welcome, everyone.
Hi from Florida.
Oh, awesome.
It's only 82 degrees here.
Oh, great. We've had rainy 2 days now.
Hello from Sudbury. Little cooler here.
Hi.
Oh, cool.
All?
Welcome.
Oh,
How's a gall wasp?
Hi from Rockville. I'm enjoying dinner and watching.
Okay, great. We get to see you. Wonderful.
Can somebody tell me how I can change my name on there to the photograph, the camera, the video?
Yeah, so go on the little dots, the 3
Yeah.
dots, and then
Yeah.
at the bottom it'll say rename. And then you can change your name. Is that what you wanted to do?
I wanted to have the video go on.
Oh, just hit down at the bottom where it says video, bottom left. Okay, well, people are filing in. This is great. Hi. Okay, wonderful. Yes. Well, one thing to remember as we get going. So one thing is we're recording, and also remember to mute. So we'll go ahead and start at 7 o'clock. Welcome to the 1228th meeting of the Entomological Society of Washington. Like I said before, we will be recording this meeting and the presentation, and then you can view it, or others can view it in our YouTube channel. So if you keep your video off, then you can remain anonymous. So if you wanted to do that. Okay, so we have a few more people filing in. That's great. So first we have a great speaker lined up, and but before we get going with that, we usually do our business. So we will be reading and approving the minutes. So Gary Hevel, our recording secretary, will read the minutes of the last meeting, which was held March 3rd. So Gary?
Sure. The 1227th regular meeting of the Entomological Society of Washington was gavelled to order by President Lourdes tomorrow on the 3rd of March 2022 in the form of a virtual meeting. 48 individuals were registered in attendance. President Chamorro offered a salute to the residents of Ukraine who are currently undergoing an invasion by Russia. Recording Secretary Gary Hevel read the minutes of the February meeting of the Society, and these were approved by the attendees. President-elect Matt Buffington noted that the annual banquet has been scheduled for early June, and the contract for the event has been signed. Program Chair Alan Norbaum announced that the speaker for the April meeting will be Miles Zhang, a postdoc working with Hymenoptera. Membership and Communications Secretary Elizabeth Young noted that there are 2 new applicants for Society membership, A.K. Curtis and Vieta Holland. Joyce Gross introduced herself as a new member. Matt Buffington mentioned that he has been busy preparing murder hornets for an upcoming exhibit in the National Museum of Natural History. Bill Murphy noted that BioQuip, an important supplier of entomological supplies and specimens, is closing its doors and will be missed. Lourdes Chamorro said she has recently used alternative suppliers from France and Japan. Gary Hevel displayed a book that has been recently published from the MONA, Malls of North America series, which treats part of the family Notodontidae. The volume is authored by James S. Miller, David L. Wagner, Paul A. Oppler, and J. Donald LaFontaine. Program Chair Alan Norbaum, introduced the speaker of the evening, Art Evans, who spoke on the development and publication of beetles of Western North America, his latest book. Dr. Evans began his program by thanking the many photographers who contributed images to the volume of Western beetles. Many coleopterists of importance worked diligently to gain knowledge of the Western beetles. Henry Fall, E.C. Van Dyke, and Frank Blaisdell accumulated collections that registered in the hundreds of thousands of specimens. Art was influenced by many individuals, among whom were Charlie Hogue, Albert Sleeper, Chuck Bellamy, Roy Snelling, and Julian Donahue. Bellamy was instrumental in getting Art to South Africa. Art has a compulsion to share information about natural history, forming an insect zoo in California, and writing many books, mostly on the subject of beetles. He presented information on Western beetles, notably regretting that he has never collected the rare bufested Juniperella myriabilis. Habitats are dwindling in the Western states, especially due to developments, and forest fires. Insect numbers seem to be somewhat stable in Western states, but with lesser numbers in the East. After the talk, many people had questions. The meeting was adjourned at 8:26 p.m.
Great, thank you very much, Gary.
Sure.
Do we have any, do we have a motion to approve the minutes? Is read. Thank you, Al. Do we have a second?
Second.
Okay, thank you.
Yeah.
Okay, the minutes are approved. I brought my gavel. Thank you, Gary. Okay, the next will be the reports of officers and different committees. So if there are any of our officers present who may want to give any updates, please feel free. Okay. And our membership secretary, Elizabeth Young, will now introduce new members, and also we invite any visitors to also unmute and say hello.
Thank you, Lourdes. We have one new member to announce for April, Daniel Wilczek as a new member. And the official members who were announced in March are R.T. Curtis and Victor Holland. And yes, to echo what Lourdes said, if there are any visitors here in attendance tonight, please feel free to unmute yourselves and introduce yourselves. We'd really love to hear from you.
Well, maybe I'll jump in. I'm Martin Hayworth, a retired physician and unretired composer with a lifelong interest in entomology. And I work very much on a sporadic basis as a volunteer and research associate in the Philadelphia Academy of Natural Sciences, helping to identify beetles and put some order, pun intended, on the rather disordered beetle collections there. So I'm here really at the suggestion, if that's the right word, of John Gellhouse, the head of the department, who notified me about this meeting. So at this point, I'm going to step off the soapbox, but thank you for allowing me to introduce myself.
Wonderful. Great.
Welcome, Martin.
Thank you. Yeah. That's great.
Any other visitors?
Hey, I'm not actually, I'm a member, but I don't come very often. My name's Jeff Clark. I created a site called gallformers.org. So gall wasps are very close to heart. Total amateur. I work in software, but I love nature. Miles and I have gone out gall hunting, so I'm really excited to hear his talk tonight.
Thanks so much for coming, Jeff. It's great to hear from you. Glad you could make it.
Glad to be here.
Any other visitors?
Hello, everyone. Can you hear me?
Yeah, we can. Hi
Okay,
there.
hello. I am Judy Kyle. I am a PhD student at BYU in Utah. And I saw this on my Twitter feed and I was like, okay, just go and see. That is why I'm here.
Well, that's great. I'm glad you found our tweet. It's really great to have you here, Judy Kyle. It's nice to see you.
Okay.
All right. Any other visitors?
Great. And you can also feel free to put on the chat if you're a bit shy. That's okay. And it's,
Absolutely. Well, thank you so much for everyone.
yeah, thank you, Elizabeth. And great to also see some recurring faces, people that come to meetings all the time. So that's great. And again, the format of doing the meetings over Zoom has just been wonderful, right? So we get to have people from all over the country and the world in some cases who are participating and joining in. So we get to see faces of the membership that we maybe never have had in our meetings. So that's wonderful. So we'll go ahead and continue with unfinished business. I don't think we have any of that right now. I'll just reiterate that we're working on our website, and that should be, you'll be receiving an email from me with some specifications and some maybe a form of sorts describing the website and what we need to do moving forward. So that should be about the only thing on that topic. So if there's any new business, I don't think there is. So we go to my favorite part. One of my favorite parts of the meeting is presentation of notes and exhibition of specimens. So if we have anybody who wants to share something with the membership, please go ahead. I know that Al had something to share. He was putting it on his screen just a little bit ago. Go ahead, Al.
Well, I don't think it will really show very well, but there's a genus Straußia that our speakers back in the fall, Andrew Forbes and Elaine Hippie talked about, but I've been rearing some, and we actually found another new species. I don't know if it's cooperating here. You can kind of see them up on the top of the vial there, but I was telling Lourdes, we collected stuff in the fall, and we found larvae in Phylogeny decapetalis, which was reported as a host of one species, but there was really no info. So we collected a bunch of stems, and these flies came out, which are not the species they said was supposed to be in there. So looks like it's a different one. So that's always fun when you rear stuff and just see what comes out. I also had a comment on the minutes, and I was trying to say that, but I was talking, I had myself muted and I was talking to myself, I guess. Gary, I don't believe that Matt Buffington would ever use the term murder hornet for the, what's the name of it? Giant Asian hornet?
Yeah, well, it's currently Asian giant hornet.
Asian giant.
But we are petitioning ESA for a new common name that is perhaps more neutral. So I won't reveal what that is now until that moves forward a little bit more. But Asian giant hornet is the current common name in use for Vespa mandarinia.
So the new proposal will not be murder hornet, I take it.
It is not going to be murder hornet. At least the proposal that I'm part of is not murder hornet.
Cool. I'll just note while we're talking that if anybody wants to say something and bring themselves to attention, if you go down to the bottom of your screen where it says reactions, if you click on that, there's a button that says raise hand, and that moves your window right to the very top of the screen where we would see you. So if you have comments at any point, please use that.
Great, we have Tina Lidwak raising her hand.
Hi. I just thought I would share an illustration, if I can screen share for a second, and just show you this cute little thing that I did for, it's kind of for Miles and the other Hymenopterist that I'm working with. Can you see it?
Yes.
Good, there. It's a tarantula wasp.
Beautiful.
So it's going to be in a paper with that Matt is spearheading with a bunch of other people. So.
Is the tarantula winning this battle?
No, they usually don't win. No. Or maybe they.
The tarantula
maybe they
always
don't.
loses.
Right. Well, put it this way. I'm rooting for the wasp. Not just today. Anyway, there you go. That's my thing today.
That's very nice. It's very beautiful. And Tina actually is the one who has done all the illustrations in my background. And this is for the laboratory that I'm associated with. I didn't introduce myself at the beginning. I'm Lourdes Chamorro, president of ESW, but most people know that by now. Anyway, I work for the Systematic Entomology Laboratory with the US Department of Agriculture, and hence the background. And Tina is our illustrator, and she's done all of these, and they represent the groups that our scientists work on. So the weevil, which is what I work on, that's one of mine right there.
Lots of Microhymenoptera too.
Okay, so if we don't have any other exhibits, then we'll go ahead and have our presentation with Dr. Miles Zhang, and Al will introduce him now. Okay.
Allie, you're muted again.
Yeah.
Darn it. How's that?
Yeah.
I'll just remind everybody again, like at the end of the talk, if you have questions for the speaker, use that raise hand thing, and we'll see you right away. And I'll just note also that we have at least 34 participants in tonight's meeting. At least that's how many Zoom connections there are, and some connections have multiple people there. But tonight I have the pleasure to introduce Miles Zhang. Miles completed his bachelor's at the University of Guelph, master's at Laurentian University, and his PhD at the University of Central Florida. He's worked as a postdoc at the University of Florida before joining the USDA Systematic Entomology Lab as a postdoc. He works primarily on the systematics and phylogenomics of parasitic Hymenoptera, with a particular interest in gall wasps and their natural enemies. And he is also the Hymenoptera subject editor for the proceedings. So thanks for joining us tonight, Miles. You can take it away.
That's good. Okay, thank you so much for the introduction, Alan. All right, here we go. Hope you can all see the screen and hear me all right. Give me something. Yeah, okay, perfect. Yeah, so the title of my presentation is called The Advances in the Study of Gall Wasp Communities in the Genomic Age. And this is sort of like my own progression throughout my field. I started off wanting to be a traditional taxonomist. I was interested in traditional morphology describing new species, but then I guess you seduced by the dark arts of phylogenomics, and now I spend more time looking at DNA, but I still do some taxonomic work whenever I find the time. So in fact, I actually participated. I was just telling Lourdes and Alan that I visited the Smithsonian for the first time as a master's student working on rose gall parasitoids. And this is when I first met Mike Gates, who is the world expert on euretomids. So this was exactly a decade ago. Now he's my boss again. So I guess it works out quite nicely. And Matt Buffington invited me to attend the ESW meeting when I was visiting. So I thought that was really cool that you guys have your own society just in DC, which is fantastic. And the last time I visited, this was 2019, I think, or I think 2019. And I was doing some more phylogenomics work. And now I'm back for the third time as postdoc. So now that finally the Smithsonian is opening up, hopefully I'll see more of you around for those of you that work in the area. But to get things started, if you're not familiar with gall wasps or just galls in general, they are essentially plant growth that's caused by another organism. Now, this can be fungi, bacteria, virus, nematodes, mites, but of course, being in the entomological society, we are interested in insects. Within insects, 6 different orders can induce plant galls. Now, this definition varies quite a bit. From this figure on the right, you can see basically some sort of leaf rolling to an invagination to fully enclosed. All of this can be considered as galls. And the reason why many herbivores decide to feed as gall inducers is because this not only provide food for their young, but also protection. Right? So, instead of just being a caterpillar that's feeding on a leaf, completely exposed to predators and parasitoids, you have a little more protection being surrounded. You're concealed. It doesn't mean 100% protection, and I'll talk more about the natural enemies associated with these galls later. And gall wasps are in the family Cynipidae. This is the second largest group of gall inducers, the largest being the Cycidomyiates, which are the gall midges. There are about 1,500 known species worldwide. Majority of them are found in the Northern Hemisphere. And they're either known as gall inducers, meaning that they make their own galls, or inquilines, which means that they cannot make galls, but instead they take over other galls and sort of modify the gall tissues. Sometimes they will kill the host in the process. Sometimes they'll leave them alone, just be, I guess, the freeloader. The majority of the diversity are found on oaks. So out of 1,500 species, about 2/3, so around 1,000, are known from oaks and other related Fagaceae plants. But then other species are recorded from roses, asters, and like Lamiaceae kind of herbs. So there's a bit of diversity there, but the majority are on oaks. And that's probably the focus of my talk today. So when we talk about gall wasps, probably the first question that we get is that how do they make these galls? And the answer is still, like even now, is we're not entirely sure. There's a lot of interesting evolutionary studies that has been done on these systems using like state-of-the-art transcriptomics to look at stage of development. But basically, it boils down to they change the plant tissues quite a bit. And in fact, depending on the layer, so the outer layer of the galls have a lot more defensive compounds, so like chemicals that will protect them, like tannin, whereas the inside actually suppresses the plant tissue, so it makes them more easier to manipulate. And there is all these novel genes that they can turn on to basically create novel tissues inside the galls for the larva to feed on. So this is an active field that is a hot topic to research, but this is not my expertise, and I actually don't like. I should know more about this, but it's not my background. I'm just sitting in the back and waiting for people to come up with exciting results, and I can just try to learn this myself. So whenever people ask me this, I have an answer other than I don't know. But regardless, you can see that there's a lot of diversity with oak galls. So here are some pictures of the galls that I took pictures of. Many of them are actually from Archbold Biological Station. That's where I did field work right before the pandemic. Actually, I left because the pandemic started and I had to, get back home. So you can see that there's a lot of diversity with the galls, and they're on different parts of the plant. They look quite different. Some have hairs, some have spines, some can secrete nectar. Some even have this hollow suspended larval chamber in the middle. So the gall's much bigger, but then the chamber is just suspended in the middle. So there's a lot of diversity in this. And the reason why we think that there's so much diversity in the gall structure, even though the gall wasps themselves look very similar to each other, is because they're doing this as a deterrent for their natural enemies. And speaking of natural enemies, there's a whole variety of wasps associated with the gall systems. So in addition to the inducers and the inquilines that I talked about earlier, you have a variety of parasitoids, and then hyperparasitoids that attack the parasitoids. And many of these are chalcidoids, but also there's some braconids and a few other critters out there. So you have this entire ecosystem that is all based off the single source, the gall tissue. So it's a really fun system to study. And of course, this is not limited to gall wasps. You could easily do this with gall midges and other systems, but galls in general, I think is really cool. And many other people think so as well. So it's been used rather extensively as a model system to study evolutionary biology, insect-plant interactions, like community ecology. But the majority of these work are actually done in the Western Palearctic, so in Europe, where the fauna of the gall wasp and their associated members are best known. And by comparison, North America and Asia are much less studied in terms of the diversity, and we just don't know nearly as much about the fauna. Which is really a shame, because if you go through the literature of North American gall wasps, you find a lot of like interest early in the 1900s. So Alfred Kinsey, the famous human sexologist, he started his career actually as a gall wasp taxonomist. He was a prolific collector. I think he collected like 9 million specimens or something. Most of it's now stored at the American Museum of Natural History, but we definitely have some here at the Smithsonian as well. Before he moved on to, I guess he gave up trying to figure out gall wasps and figure human sexuality is easier to deal with. But regardless, he's made incredible contributions to our field. And you can see on the right, this is figure of his idea of a phylogeny of how these different genera are related to each other. So this was published, I think, in 1920, so like over 100 years old. Another person is Lewis Weld, who worked a little bit at Smithsonian. He actually lived in Arlington, where I live right now. But he's independently wealthy, so he spent a lot of his time just going around and collecting galls or getting people to collect galls for him. And he privately published a bunch of these articles on like the gall wasp fauna of the America. Like the different American regions, such as the Pacific Slope, the the Southwest, and then the Eastern United States. And we actually still use a lot of his references to this day because it's still the best resource that we have. But unfortunately, there's not a lot of focused research on Cynipid taxonomy as a whole in North America. And where the majority of this research done is in Europe by Europeans. So here's George Melica, crowned himself the king of the galls. So he made a hat out of galls, which I would love to have one. So he is the world authority on gall wasp taxonomy. He's based in Hungary. And another person you probably have heard of is Frederick Rundquist, who is probably best known as a Bayesian phylogeneticist. If you ever used the program Mr. Bayes, he is the lead author on that. But he is also instrumental to our understanding of gall wasp systematics and taxonomy. So getting into the taxonomy, currently by the Rundquist, he recognizes 12 tribes, and then a more recent paper published by Matt Buffington and Bonnie Blamer recognizes 8 tribes. But regardless of how you split it, this fantastic key that was just published 2 years ago led by Matt Buffington will get you down to the tribal level. So it's fantastic. It's free, open access, at least I think it's open access. Go check it out if you're ever interested in keying out Cynipoids. But beyond the tribal level, it gets a little problematic, and the majority of the problem lies within the gall or the oak gallers, which are in the tribe Cynipidae. Like I mentioned, this is the majority of the diversity. So currently there's about 51 genera, but there's a lot of changes that happened in the past 10 years or so. In fact, I think just last year alone, we have maybe 5 or 6 new genera being like just popped up. So it's constantly changing. And it's not because people have done people have done a bad job of classifying them, but it's because they're really hard to identify. And part of this problem is due to this bizarre life cycle that they have called cyclical parthenogenesis, where they switch between the sexual generation and the asexual generation. So in the spring, they have both males and females. They make one kind of gall. And then in the fall, they make a different kind of gall, but they only have females. So here's an example of one of these life cycles. So on the top, you have a sexual generation, which have both females and males, and they make sort of like an underground root gall on live oaks. This is the genus Ballonochnema. And then here's the asexual generation. It's the same species, but you can see it looks very different. In fact, the one on the bottom cannot even fly. They have these little twisted brachyporous wings. And so all they do is they dig, they come out of these galls and they go straight underground. They use these big tarsal claws to dig into the soil, and then they'll go and start inducing these potato galls. So you can see from a morphological taxonomy perspective, this is a nightmare. So like they look completely different. They're found in a different part of the tree. So in the literature, you see that many of them are described under different names, and sometimes even in different genera. So it's really problematic. And of course, it's really hard to link them together, at least back in the days where you need detailed ecological rearing, and which is often really hard to do for anyone that's tried rearing galls. But of course, nowadays we have molecular tools that we can link different life stages, and also you can try to link these two life cycles. We often call it closing the life cycle, right? Because often we know half, but not the other. So we're trying to match this life cycle up for many of the species that we know. So in terms of challenges, working with cynipids as a whole, we definitely need accurate rearing records. And if you dig through the literature, including whales, we actually notice there's quite a bit of mistakes, which unsurprisingly, when you rear hundreds, thousands of galls, you probably get things mixed up here and there. But you know, these things persist through the literature, and then we just keep perpetuating it by not double checking these records. And that's definitely been problematic. We're trying our best to correct that. And this of course includes the accurate host. Oaks are notoriously difficult to identify. They're hybridized like crazy. So it definitely makes things a little harder. But also looking at the phenology and even the gall descriptions themselves, right? So just if you just collect the gall in a Malaise trap or something, they're often, a gall wasp in the Malaise trap, they're often really difficult to identify because we don't have the associated information. So having this ecological information is really important in many of the new species descriptions, but also in how we study other things, which I'll talk about, such as the parasitoids and how they choose which galls to attack. Luckily, there's some really good resources out there. If you live in the western half of this country, you have this excellent guide that's been recently updated. This is the Plant Galls of Western United States. This encompasses more than just oak galls and cynipids in general, but it's a great resource for anyone that's interested in this. But also now with iNaturalist and this accompanying app, Seek, you can go out and point to something and identify it, right? They're not great for many insects, mainly because insects are too small for iPhones to detect. But for galls, it's actually not too bad because it's sort of like looking at plants, right? iNaturalist is really good at identifying many genera of plants. Oak, not so much, but it works quite well. And in fact, there's a group of dedicated naturalists out there. If you go on iNaturalist, you can see they have these projects that people created. So here's a galls from North America that people have made. And of course, this encompasses, you know, the Eurasta, the gall flies, the midges, what have you, all kinds of galls. So it's a great place for people to contribute, right? Anyone can upload pictures and then they get curated by volunteers. And some of these people are such as Jeff, who introduced himself earlier, are amateurs by training, traditional training, but I bet he can out ID any of us in terms of the local fauna and the trees. It's been great going out and learning about oaks with Jeff. So, and of course, speaking of which, like the gallformers.org is a website that's led by citizen scientists, and it's a fantastic resource to not only have the distribution records, because we don't actually have a formal database for cynipids. So this is the best resource that we have. It's great to just quickly look up the literature and then find associated distribution. And even they have these codes that for undescribed species. So we can actually, you know, it's like, oh, okay, so this one is new. So if we ever see it, we can sort of link it to what is known, and it's often linked to some iNaturalist record. So it's been great to use this as a resource to find new distribution records or even these new species. And this currently encompasses US and Canada. So if you're interested, go take a look. I personally love it. I can't imagine what I would do without this nowadays. But so it can lead to so much more. So here's an example of urban biodiversity, right? So here in North America, we often think it's like, oh yeah, we know many of the groups quite well. Cynipids, of course, is not one of those. And so here's an example where one citizen scientist in Texas, she found these galls on an endemic oak, Quercus lacei, only found in a few spots in Texas. And she collected these and she reared this out and then done these photographs of these live specimens. And she sent it to me. And it turns out it's a new species. So we're in the process of describing this. But even before this description is done, she's already collaborating with Erica Newman, who's a scientist at one of the University of Texas Permian Basin. And her students were interested in this. So they collaborated, they went out to look for another endemic oak, and they already found more species as well. And so, and this also includes one of these rose gall wasps that I was also interested in. So there's so many out there probably that awaits discovery. And I love collaborating with students and community scientists rather than, just other taxonomists, which is a lot more fun because like local experts have the knowledge and they're physically there all the time. So it's better than any, collecting trips where you can only go maybe a few weeks, a month at a time. So, that's one of the challenges, and hopefully we can circumvent this by, using these these publicly available resources. But we also have the problem of higher phylogeny. So currently the generic limits for cynipids, especially Cynipidae, is just a mess. Many of them are para or polyphyletic, and we definitely need better ways of identifying them. And the taxonomic revisions, right? Once we know what we're dealing with, it will be easy to do taxonomic revisions because currently you probably key things to like Androcus or Caloritis, which are like the two big genera, both of which are polyphyletic. So it's meaningless. And you can't really do a taxon revisions when you don't even know where to start. So hopefully we're in this process of closing the life cycles and doing these revisions. So, and this leads me to this paper right here. So this is what we currently, or at least a while ago, this is what we know about the phylogeny of cynipids. This is by Frederick Rundquist, led by him and a few other authors. And this is from 5 genes plus morphology. Right? So, and here they use Ebaliids as the outgroup, which is a macrocynipid, right? So it's always thought that Phygidids and cynipids are the microcynipids. They're closely related, and all the other macrocynipids are more distantly related. So they chose Ebaliids as the outgroup. And this is the tree that they've generated. Here you can see that Cynipidae is shown to be monophyletic, right, colored in green, and then the inquilines have evolved multiple times within this group. But this is a secondary switch. So what this, in this tree, at least, they've shown that gall induction have only evolved once, right, as the ancestor of cynipids, because Phygidids, which is the sister group, are parasitoids. So like, this is what we thought we knew, and everything's like, oh yeah, that's just, that seems normal. But with the advances of genomic technique, we're getting way more genes, right? So now we're capable of sequencing thousands of loci across genome at low cost. You probably have seen these papers by Misov et al., the science paper on like the insect phylogeny, or if you work with Hymenoptera, this Peters et al. Paper, it's a tree of Hymenoptera. All of this is generated using transcriptomic data. That's the same kind of data that was used in the gall induction study, but they can also use this to build phylogenies, right? Because you're using transcriptomics, you essentially have thousands of protein-coded genes to build trees from, and this is a much bigger data set than the 5 locus gene tree that I've shown you in the previous slide. And this was published actually a year before. So you can see how fast the technology is moving, right? Within Hymenoptera, many of us recently have adopted this method called ultraconserved elements, or UCEs for short. And essentially what it does is you design these little probes to capture these targeted sequences across the genome. They're called ultraconserved because they can be found across of deep lineages, like between humans and all kinds of mammals. In fact, tetrapods, you can get all kinds of ultraconserved regions. So by targeting these and capturing the regions next to it, which are a lot more variable, and those are where the phylogenetic signals are. And so you can build phylogenies using thousands of loci. And that's different than the transcriptomics, because transcriptomes, you need really fresh specimens, but here you can work with degraded specimens. And they have such high success rate with degraded samples. Now we're calling it like, right, everything is something omics now, genomics, transcriptomics, now they're calling this museomics, right? Because you can use museum samples to do this, which is really exciting. This opens up, some of these older specimens that we can sequence. And you can, of course, combine with any of these resources that I've talked about, whether you have genomes, transcriptomes, anchor hybrid enrichment, you can combine it all together. So it makes it really versatile. And it is backwards compatible, at least the probes that we're using in Hymenoptera. So a lot of these legacy markers like CO1, ITS2, 28S, these genes can also be captured in the same way, which actually works very well for me, who works on a lot of chalcidoids. Like sequencing CO1 in chalcidoids is just really painful. And now I'm to the point where it's easier for me to just fish out the CO1 from the UCE samples than try to bang my head against the wall, or do like, I don't know, essentially voodoo to try to get some CO1 sequences out of chalcidoids. So it's been really versatile to work with. And in fact, here's the paper that was published 2 years ago, led by Bonnie Blamer, who was previously a postdoc at Smithsonian, along with Matt Buffington, Sean Brady, and Diedrich Otzek. And so here they've done UCE phylogeny for cynipoids, right? So we have over 1,000 UCE loci, mostly from museum samples. And this time they use platygastroids as the outgroup, right? So other studies have shown that platygastroids is the sister to cynipoids. So it makes sense to use this as an outgroup. But what they got, the tree was drastically different than the Rundquist et al. Study. Here, it shows that Cynipidae is actually polyphyletic, no longer monophyletic. And a few of these genera shown with these blue arrows are traditionally these tribes within Cynipidae actually falls out in many different parts of the tree. So they are essentially, they're not really Cynipidae, and they need to be elevated into their own families. And this change fundamentally changes our understanding, right? Because now Gall induction have evolved multiple times rather than a single time like we previously thought. And I started my career working on the rose gall wasps, the Diplolepidini. So now I guess they're their own family, which works for me. So, but with these new methods, it's really exciting for someone that like playing with DNA as much as looking at specimens, right? So here's an example where we describe the new species, a tiny one from Neuroterus from Texas. We named it Valhalla, and we described this species and we sequenced its entire genome in the process, right? So as far as we know, this is the first time anyone has done something like this, describing a species and its entire genome. Of course, when you have a whole genome, you can do all kinds of analyses like comparative analysis, but also in this case, we are able to fish out these UCE sequences. We can add it to the phylogeny made by Blamer et al. And that's exactly what we did here. So what we have shown, again, this is something that we've known for a while, but again, using UCEs, we're able to place these taxa accurately. So the Nearctic Neuroterus shown in green is actually very distantly related to the Palearctic ones shown in orange. So this genus is originally described in the Palearctic. So probably all the North American Neuroterus need to be moved to a new genus, which hopefully at some point that we need to do. And this is just an example of like slowly we're figuring out the phylogeny of Cynipidae bit by bit, because many other groups are still remain problematic. And here's a study that we collaborated. Many of us are on this paper, and it's actually led by Andrew Forbes's lab, which Alan mentioned. His lab also works on a variety of other systems, but he's always been interested in gall wasps. So with his newly Dr. Anna Ward, who just defended, I think, her PhD last week, they published this paper. We're using UCE phylogeny of newly generated sequences of a bunch of the Nearctic species when combined it with the Blamer et al. Paper as well. So this is the largest UCE data set for this family, or for this group to date. And what we have noticed is that this group have shifted hosts many times, right? So we're looking at how they shifted within the phylogeny of the oaks as well. So between the different subgenera and the sections of oaks. So they actually jumped around quite a bit, and they actually moved multiple times from the Palearctic to the Nearctic and going back and forth. So this is pretty exciting for us because now we can sort of look into the evolutionary origin of how they moved around and how they shifted in relation to their hosts, because previously this type of study is only done in Europe, where the diversity of oaks is much lower, whereas here in North America, we actually have a much higher diversity. So this changes the narrative quite a bit. So it'd be great to also somewhere down the line include, the Asian fauna as well, to compare, how it changes depending on the oak fauna and in relation with the gall wasp themselves. Okay. And in this process, we sequenced a bunch of these samples using UCEs, and we actually noticed that there's this many unresolved taxonomic issues that we can hopefully address. This involved closing the life cycle. So here's two species, currently named as two species, but genetically they're identical. So this is probably the spring and fall generation of the same one. So this is an Amphibolyps. And here's another pair. One is wingless, and this is currently in the genus Zopheroteres. And then on the one on the bottom is Triocosmus, which are winged. And so these two are actually the same thing as well. So we need to combine this together. You can see morphologically, they look very different from each other, but now we know it's like, oh, actually, maybe this genus is just the alternative generation. On the other hand, we have something like some subspecies. So Kinsey, even though Alfred Kinsey, who named a lot of these species, he's a notorious splitter. So everything is a new variety, new subspecies, is really frustrating to work with. So here are two Neuroterus species that make those little jumping galls I showed earlier in that cartoon. So one is found in California, one is found in Texas. The one in Texas is currently just a subspecies of the California one, but then our genetic data have shown that they're actually pretty distantly related, still within the same genus, Neuroterus, but they're two very distinct species. So this will need to be elevated. So there's a lot of taxonomic work that we highlighted. Hopefully, I know I won't have time to tackle the entire family, but hopefully we get more and more people interested in this group, that we can sort of address all of these taxonomic problems one at a time. So that's sort of like the first part of my talk, just focusing on the gall wasps themselves. But like I mentioned before, I actually started my career working on the parasitoids because I like the parasitoids more than just the herbivores. They're more fun. And there's a variety of parasitoids associated with gall wasps. And here's a collage of some of the things that we reared out of a single gall. And you can see there's some like ants and Socotrans in here, which seems weird, but they actually utilize these older galls. They live in them. And so there's a lot of interaction that we don't really know once we start rearing more and more galls. But the majority of the primary parasitoids, at least, are chalcidoids. And I've always been interested in eurytomates, and this is what I'm currently working on in this genus Sycophila. Now, Sycophila, in North America, they're mostly associated with oak galls, but in other parts of the world, they're associated with figs and a few other galls as well, made by chalcidoids. And even if you're from the Southwest, they're on the yucca moss. They are parasitoids of the yucca moss as well. But the last time the only key we have in North America was published 99 years ago, and then they recognized, I think, 40 species. Most of the differentiation is based on color and wing pattern. You can see that often these species have really seemingly distinct wing patterns. So I thought it was like, oh, this seems like a fun group to tackle. They're prettier than the Eurytoma, which are just mostly black. How wrong I was. A lot of them have really wide intraspecific variation, right? So these color patterns changes pretty quickly. So then they become really unreliable. Where does one species end? Where does another species start? Right. So for this study, we used, again, in collaboration with Andrew Forbes's lab, and using some of the Smithsonian material, we sequenced as many as we can find. We ended up with 35 morpho species. And this is mostly done using the traditional DNA barcoding CO1, but we also used a subset of UCE data as well, just to corroborate the findings of the CO1. And we did a whole bunch of different species delimitation techniques. For the most part, it seems to like these clusters are okay. Whether they're true species or not, sometimes these methods tend to oversplit, but that's for another debate. But regardless, there's more than what we previously thought. And many of them are actually specialists. They're focusing on just one or just a few types of galls rather than being, a generalist that will attack many different galls. In fact, we only found one true generalist that attacked, I think, 12 species of the galls out of all the, I think we had around 50 types of galls. There's only one that was a true generalist. So that was pretty exciting. But we're also interested in more than just that, right? So we want to know how these Cycophila are picking which galls to attack. And this is where all these gall traits come in, right? So this is why I was mentioning earlier, it's good to have the descriptions with the galls, because then for this study, I looked in the Smithsonian gall collection to characterize many of these gall traits, and then combing through the literature to find whether to describe these are as like fleshy, woody, textured. And quickly I realized that these terms are also quite ambiguous, and we need to update this, and maybe as a field, we need to have standardized terms to make it more feasible to do these type of studies. But we got to start somewhere. So I tried my best to categorize them. And then we plotted all of these traits on sort of like a PCA, right? So this is called correlation matrix, so PCOA. So you basically have all these multivariate traits, you squish it down to a 2D plane. And then we mapped all the Cycophila, the parasitoid, onto this gall trait space to see how they are interacting with the trait space. And what we found is that actually distantly related species are converging on similar traits, right? So instead of being a strict co-evolutionary scenario where as the gall wasp, as the parasitoids diverge with their host, they will just keep following their host's gall traits no matter how they evolve, they're actually focusing on similar traits. And in this case, it's galls with less external defenses. So you see that here's two pictures of the galls that they are attacking. They don't have a lot of spines or hairs to associate. Maybe that this is just easier for them to oviposit. Right, but of course, these galls are not evolved just to keep out Cycophila. They have to deal with a whole group of parasitoids. So it will be great to look at this as a whole picture to see, maybe some parts of these traits are really good at keeping out Pheremis, but not Cycophila, and vice versa. So that is something that we are hoping to do in the near future once we have robust phylogenies of many of these parasitoids. And of course, I can't do this by myself. Luckily, there is a renewed interest in North America for gall wasps. But of course, some people have been doing this much longer, such as Matt Buffington, who's been, even though he works mostly on Phygidis, he likes to dabble in Cynipids once in a while. But we have a new group of people focusing on different things, such as evolutionary biology, ecology, but also, of course, taxonomy as well. And it's been great to working with many of these people, or in the future, I'm hoping to collaborate with them to answer some of these questions that we're really interested in as a group. So here's another example of one of the gall wasp parasitoids that we've discovered. So if you just focus on the top half of this figure, this is a regular gall wasp, lay its eggs, make a gall, the gall it emerges regularly. So end of story, that's normal. But if they were attacked by this tiny little eulophid called Euderus set, shown in green here, instead of leaving the gall, the parasitized host will chew a tiny hole on top of the gall, but they wouldn't leave. It's not big enough for them to exit. So it just chewed big enough you can see its head, and then it will use that head to block the hole, and then they will die right there. And then afterwards, this parasitoid actually eats its way out of the body of the dead host and then emerges out. And the reason why they manipulate their host into chewing this whole is we think it's their jaws are not strong enough to chew out of the galls themselves. So they somehow get their host to do it. How do they do this? We don't know. But you often in the field, you'll find these galls have these little like heads popping in there. It looks like they're just like looking out from out the gall, but they're actually dead. So it's kind of creepy, but also really cool. And this is just one of the many species of the parasitoids that we're just now discovering. So this has actually caught the attention of a few of the mainstream news. So Ed Young, if you're a fan of his on The Atlantic, he wrote about this, which I can check off my bucket list as something I always wanted, but also we were being covered by a few other sources as well. So like my colleague in Texas, they found these love vines, the plant, parasitic plant, is actually parasitizing the galls. They're sucking the nutrients and killing the wasps as well. So there's a lot to be discovered, and people tend to find this interesting. So I think it's a great way to, use this as a tool to educate the public about the diversity of insect, diversity of parasitic wasps in general. And this study actually just came out yesterday and by the group at Penn State led by Andy Dean. So this is Cococynips riley, and then Decidua. These galls have like this little fleshy top on the galls. And what they found is actually this is used to attract ants, and they're using this to disperse the galls, right? So the ants will eat this little part, and then they'll often take them back to the nest, and then this is how the galls gets dispersed. And this is actually, you probably have heard something like this in other plants as well, but this is actually the first case that we as far as we know, that galls are doing the same thing. So it's really cool that we can still have these really neat natural history discoveries, even in our backyards where these galls are often commonly found. So it's really exciting for someone to be working in this field right now to still be able to do these type of studies, but also do these deep phylogenomic studies. And for someone that's interested in both cynipids and chalcidoids, it's great because I also ultimately want to see if there's any parallels here, right? So this is the study I published with Mike Gates and Matt Buffington. So this is one species that is a mistake in the literature. They always said that it's like, oh, there's one species cynipids that's on Smilax, which is the only monocot plant. Every other cynipids are on dicots. Like that doesn't seem right, and it's only found in only found in southern Florida. So Matt and Mike have done some like collecting down there in the rare dust specimens. We did like some multi-locus phylogeny, and then we did the description. So it turns out this is actually a eulophyte that made the gall. So here's another drawing by Taina, which is included in the publication as well. So there's no cynipids on monocot, and this only took us like 100 years to correct this mistake. But it's definitely important, and because there's a lot of chalcidoid gallers, especially in the global south. Instead of cynipid diversity, most of the gallers, Hymenoptera gallers, are chalcidoids as well. So you can see some of these. Some are pests. We have here like on blueberries, the Pteromalus hematus nubilopennis. Of course, the fig wasps. They're technically galls as well. Right, so they've also evolved multiple times within Chalcidoidea. In fact, as far as we know now, it's more than 15 times. So now that we know cynipids have also evolved multiple times, or cynipoids evolved gall induction multiple times, it'd be great to do these comparative analysis to see if there's any similarities on how they make the galls, right? Whether evolution repeats itself, or is this something completely novel ways of making galls? So it's very exciting time, I think, for me. So with that, just want to wrap up. So we've come a long way since Alfred Kinsey, from that tree he made in 1920 to Blamer et al.'s paper in 2020. But there's still plenty of unanswered questions, and this ranges from natural history to very I guess state-of-the-art genomic studies. But in order to do any of this, we need these robust backbone phylogeny, because right now we don't know what we're doing. And we need to revise this, right? So taxonomic work is fundamental to do any of these evolutionary studies. And these new target capture methods seems to be really promising. And I'm looking forward to exploring more about this world of galls using these methods. And of course, I can't do this by myself. Like I mentioned, I have many collaborators. This includes some of the mentors, collaborators, including citizen scientists and various funding resources. So with that, I'll be happy to take any questions. Thank you.
Thanks, Miles. That was great. I could just maybe start you off with a question. I was just curious, like, how stable are the Hymenoptera phylogenies that are being generated from the genomic methods? Like last Diptera Congress I went to in 2018, people were looking at different methods, and there was good agreement on the well-recognized clades, but like a lot of the problems still were problems. People were getting different answers. So I was just curious, like, how it's happening. And then one more thing, can you tell us more about those jumping galls? That was pretty cool.
Yeah. Okay. So first part, yeah, that I think Diptera had more problems than Hymenoptera as far as we know. But yeah, if you go back deeper into the phylogeny, there could be some mistakes, but I think for the most part, we kind of settled on at least the superfamilies. Are you talking about between families, or I don't know where the Diptera trees are? Is it the acalyptrates that's problematic, or?
Yeah, well, in Diptera, there's a lot of questions about the early branches within different groups, like within the Brachycera and early branches of what was traditionally called Nematocera, and like within like what was formerly Cyclorapha, there's questions about how the branching occurred.
Yeah,
I'm
friends with a lot of Diptera, so I hear about their gossip, their family level gossip. Yeah, so I think a lot of it also depends on the analytical method. So now we have more genomic data, we need to figure out how to analyze these data better. And like I'm involved in this completely different project on ant phylogeny. We're looking, we have genomic level data for all of ants, and they also have some problematic groups there. And depending on how you filter the data, how you analyze the data, you get either monophyletic or paraphyletic. So that's what we're trying to figure out. So at these deeper nodes, you have these discordance, and it's caused by some unknown, like we try filtering in specific ways. You can sort of minimize that problem, but there's still some evolutionary process that we don't really understand as a field why there's discordance. So it's not like a Diptera specific problem or so Hymenoptera, I think for the most part, we have the family levels figured out, but as a field, we need to spend more time thinking about these type of questions, or like what's the best practice, how to analyze these type of data so you get like the repeatable answer, or at least figure out why there's this deep introgression or whatever that's causing these problems.
Yeah, I know in Diptera, like one thought is that maybe these problem areas, just there was rapid evolution, and there was that whatever signal there originally was has been swamped out by all the convergence that's occurred. So I don't know if that's the same situation.
There's some analysis we can look into, like in ancient introgression. You can sort of see if there's like weird mixing or like rapid evolution. But yeah, like I don't think there's a best method yet. I think we're always waiting for the vertebrate people to figure it out, because they figured out all of their taxa, so they can focus on the methods, and then we can apply it to our hyper diverse groups and see what the hell's going on here. So I don't know. I guess this is Keith Bayless needs to figure this out or something.
Yeah, right.
Yeah. So as for the jumping galls, so yeah, so sort of like the Mexican jumping bean. So it is the larva that's actually hitting against the gall wall. So and they're doing this to, I guess, to get out of the sun, or in an optimal area where they can overwinter. And I think well, like I've never seen it myself. I'm hoping to see it someday, but maybe Matt knows more about it. Like they're just like rain down from the skies in like parts of California, and you can pick them up by the spoonfuls.
Yeah, they're extremely small, and when they massed out of the tree by the millions, I'm not kidding. And you look down on like a driveway, a flat surface, it's all just these little tiny microscopic, like poppy seed size objects bouncing around, and they like to get into cracks and crevices. They have as Miles said, they're looking for an optimal overwintering site, and that's theoretically what they're trying to do, is trying to bounce around basically until they get themselves into a little niche.
Cool.
Thanks, Matt.
Any other questions?
Okay.
Well, great talk, Miles. I have two. One is a pretty easy one about your photography. I think some of it you do yourself.
Yeah.
Maybe you can speak about that. They're awesome. They're really great. And then the one where you described the new species and you found the genus to be polyphyletic. Why not just describe the new genus in that paper, or were you kind of still waiting for more evidence to just erect a new genus?
Right. Yeah. We don't. So let's start with the phylogeny question. So yeah, morphologically, at least I guess people haven't been able to find any like reliable characters, and I think everyone's kind of hesitant. It's a big genus, like that's found in Palearctic and Nearctic. I think there's a paper that's coming out soon on the global phylogeny that will implement a lot of these changes. So I guess I'm hopefully leaving it to those people to figure this out. Like with George Malika, who spent his life looking at these things. I'm just a newcomer by comparison. So yeah, I think it needs to be done, but and it's been discussed many times, but everyone's kind of hesitant to do it. So we just need the big wigs to agree, which might be harder. But now we have really solid phylogenomic data. They're like, okay, fine, so be it. So hopefully soon. As for photography, so this is actually a method that Alex Wild uses. So I went to a few of his bugshot courses. I don't know if any of you have been to it or heard of it. He teaches it with a few other people. So this is actually a white box. So you build like a paper cardboard box, and then you point a flash directly upwards. And so the light hits the top and about like diffuses down really nicely and evenly. So you can even put a plexiglass on the bottom, and then you get like a really cool shadowy effect. So yeah, if you Google like white box, maybe Alex Wild, you probably find something like that. It's a really fun way of doing, the white, and you can change the background, of course, but of course in situ it's a bit harder. But luckily gall wasps are not the most fast moving for the most part. Makes things a little easier.
I also like that you image the galls. They're like little works of art. They're very beautiful. Yeah.
Makes it more fun and easy to input into the manuscript. It's already in white background. Yeah.
Miles, I know one thing. You should be very proud. Your work got in the Atlantic. Really?
Work on the what?
Your work got into the Atlantic?
Yes.
You should be very proud.
Yeah, I am. I'm a big fan of Ed Young, who writes like popular science really well. So I was like, yeah, I was quite happy about that.
Yeah, I wonder how many entomology articles have been in the Atlantic. I don't know, but it's a great magazine.
Yeah. I mean, there's so many cool things that we just need to discover. And then I think selling it, my collaborator is always really good at, doing these interviews and other things. So it gets into these articles. I'm like, oh, why didn't I think of that, right? I'm just focusing on the specimens and like, okay, cool, next group. But where they're like, hey, everyone, look at this. It's cool. And so I definitely need to work more on the SciComm front of taxonomy.
Go ahead, Jill and Warren.
Hi, that was a great talk. I was really fascinated by those eliosomes on the galls. I mean, I don't know if you call them that, I can't remember, but I studied ant dispersal of spring ephemerals for my master's years ago, and I just think it's the coolest thing ever. And I was like, oh my God, they're on galls? I was just amazed. I had never heard of that. So is that has that been known for a while and I'm just out of touch, or is it a newer thing? And is it, being studied and,
So apparently people have observed it and hypothesized it a while ago, but this paper was literally published yesterday. So this is, as far as we know, new, right? So, and it turns out that there was one group, I guess, working independently, like two groups figured this out. I was like, oh, wait a minute, this looks like, like it's for ant dispersal. And then they so that paper is great. It's an American Naturalist. They've done studies where they cut off that fleshy part, and then the ants are no longer attracted, and ants are attracted to only the fleshy parts. So they've done a bunch of experimental studies as well to figure that out. So yeah, it's definitely super cool. And some of the other ants, like I said, they live in the older galls as well. So there's a lot of interaction with ants. Some secrete the nectar, so you get ants on there as well. They even have, I think, scale insects that they're farming inside the gall. I've cut open galls and they have that in there. So there's a lot of these things that just people haven't been looking more into.
Yeah, that'd be really interesting to learn more about, because with the spring ephemerals, the eliasoms actually attract the ants to kind of carry the seeds, but they don't need to carry a whole gall away. They just are being fed, and I guess that's to encourage them to stay around the gall. Like, I was trying to figure out, why they're there.
Yeah, I'm not sure. I read the study quickly. I was like, oh, I should put this in there, but maybe
I'm
I
glad
should.
you did. I'll
Yeah,
look for
go
that
check
article.
Yeah.
Thank you. Okay.
No problem. Hey, Matt.
Great talk, Miles.
Thank you.
I love the topic, of course. So that was very cool. So something that when Bernardo Santos was a former postdoc at Smithsonian, and he's interested in the morphospace evolutionary questions regarding ichneumonids, that they seem to be like almost quite literally boxed in. Their mesosoma, or what other folks call the thorax, that there's only like so many permutations, even across 20,000 known species and maybe 100,000 unknown. It seems the same sort of themes are going back and forth. And so I talked to him a lot about gall wasps, because they appear to be on the same scale of diversity, species-wise, as Phygitids, yet their morphospace is completely confined. Like and I think it's literally confined. Because they exist in a gall, there's only so much space there to manage. They're typically spherical. The wasps are typically spheroid. And the same is very true for the eurytomids. A lot of them have this same kind of, like, you can't really tell them apart. They're kind of restricted in their space. And I think that's a really fascinating, while it's problematic from a taxonomic standpoint, we're used to looking at external characters and being able to, I keep bugging George, why the hell isn't there a key to the genera of Cynipidae? Well, that's not trivial. Like, we could do it for the rest of Cynipoids, but we can't for the Cynipidae. And I used to think they just weren't work hard enough, but I think this is really a very difficult question. And what I'm hoping is the work that you're presenting will provide the necessary means for a sort of reciprocal illumination, where maybe we can go back to the specimens, and lo and behold, there are characters that are repeatable, diagnosable. They may not be super obvious, but I'm hoping there's something there, because it would just be a real shame if we describe all these genera, but it's just, you have to do all the sequencing in order to find out what it is, or have a complete life cycle rearing record,
Yeah, I think now that, like, at least Yuli's group, they've gone back and looked at a few, it's like they can find more characters. So yeah, it's totally the reciprocal illumination. Just when you're presented with everything looks similar enough, it's hard to, without any guidance, it's hard to find characters that's reliable.
Wonderful. Well, any other questions? Okay. Wonderful. Well, thank you so much.
Thank you.
Yeah, great job, Miles.
That's
Sorry, that was
great.
my dog that barked earlier.
That was a really good talk. Thank you. Thank you.
Yeah, yeah. Okay, so I'll just quickly say that our next meeting, well, our meetings are the first Thursday of every month at 7 p.m. and we'll continue with the Zoom style for at least for the next one, and then we're going to try a hybrid for our June annual banquet, just like we did last year. See how that goes. And now that the museum has opened back up, we'll try to have in-person meetings, but also continue with the Zoom, because I think it just has been a wonderful tool. And so Al, did you want to tell us who the next speaker is, or?
Oh yeah, next month it's going to be Yvonne Linton. She's a mosquito expert, works for Walter Reed Biosystematics Unit. So I'm sure it'll be a good talk.
Yeah, great. And then you can watch the talk again if you like on YouTube, and it'll be posted there. So thank you again, Miles, and thanks to everyone for attending. It's great seeing everyone, and great questions too. So do I have a motion for adjournment? Okay, I see. Okay, second. Okay. And with that, we adjourn the meeting. Thanks, everyone. You can go ahead and hit the unrecord or
Oh
whatever.
yeah, stop recording.
Yeah.